Welcome to part 2 of our video on cellular respiration and ATP. Now, I didn't mention this in the last video, but this is a mitochondria actually, and this is where most of cellular respiration is going to be happening. And yes, while it does look like poop, it actually has a wonderful structure that allows it to do the job it does. And in fact, we will be making references to that purple squiggly thing inside, which is actually going to hold the matrix or lumen of the mitochondria, the inner space. Now, as you marvel at this piece of work, just keep in mind that the cell is really like a factory, right? It's constantly running and producing things, and cellular respiration is one of the best personifications of this factory analogy, because in reality, cellular respiration is like a long assembly line where things are being changed and manipulated, and eventually you just get a lot of energy out of it. So, with that said, let's break it down to kind of what we've been learning so far, not only in this unit, but in previous units from last semester. So, humans, or animals, or anything that really needs energy, will take in food. And so let's actually use the example of starch. So starch, which can be found in a lot of carbohydrate-rich products like spaghetti, like pasta, bread, rice, and so on. So starch is actually going to be one of the things we eat and eventually digest via the process hydrolysis. Remember that, hydrolysis. In fact, we actually did that lab with salivary amylase, where we broke down starch into smaller products so that when we ran the Lugol's iodine test, it didn't turn purple. Well, anyways, so starch is broken down to simpler sugars or monosaccharides like glucose. In fact, alpha glucose. And so glucose, a monosaccharide, is going to be our main player in cellular respiration. So if you have this glucose right here, this glucose is actually eventually going to become what provides us with most of our energy in our body, or ATP. Adenosine triphosphate, remember how that allows us to do mechanical transport and chemical work. And this entire process actually is going to be sort of the crux of the matter. This is going to be my cellular respiration, and definitely choosing this tool to write was a mistake. Respiration. Okay, so going from glucose to ATP, this is going to be, I am never using this writing tool again. Glucose to ATP is going to be cellular respiration. Okay. Alright, so last time we talked about what was being oxidized, what was being reduced. Leoger, lose electrons, you are oxidized. Gain electrons, you are reduced. And so this is an important idea because glucose, or C6H12O6, don't want that. Glucose, or C6H12O6, is going to actually lose electrons, and then O2 is actually eventually going to gain those electrons to become reduced. And the reason why we care about where the electrons are going is the movement of these electrons are eventually going to be extremely important for running the machine that makes us ATP. So think of these electrons as little like tokens that you put into arcade games that'll eventually give you tickets, and the tickets are like the ATP that lets you buy stuff, like a chessboard at Chuck E. Cheese's. Okay, that analogy is quickly breaking down, so I'm going to move on. Now, NAD+ is an extremely important molecule. It's actually called a coenzyme. A coenzyme is not a protein. It is an organic molecule, though, that does help proteins and will bind to proteins. And so you don't have to memorize this incredibly complex chemical structure. But NAD+, if you see here, this is all NAD+. NAD+, what it's going to do is it's going to take in electrons. Okay, so it actually holds these electrons, and when NAD+ takes electrons, it becomes something called NADH. So a shorter way to see this is just NAD+ plus electrons, and with the electrons come some hydrogens, makes NADH. So really what's important about this entire thing is that NADH is simply an electron carrier, or an electron shuttle. So just think of it as like a coin pouch. if coins are the electrons, NADH is a coin pouch that will carry around those electrons. You don't want to roll your coins around Chuck E. Cheese's to get them to the game stations, right? You want to hold them all in one place. So NADH is that coin pouch that's going to hold the coins, or the electrons, that eventually allow you to cash in for the ATPs. Alright, so importantly, NADH and FADH2 are our two coin pouches. Think of, in a way, NADH as being a slightly bigger coin pouch. That's my coin pouch. And FADH as being a slightly smaller coin pouch. And now, this analogy is simply used for the purpose that NADH is going to actually allow greater yield of ATP from the electrons that it carries, and FADH is going to allow slightly smaller yield of ATP from the electrons it carries. But both of these molecules are going to be electron carriers, really, in that they're going to be eventually dumping those electrons somewhere to allow ATP to be formed. Remember, our goal is to get this molecule, ATP. Okay. Speaking of which, it should be interesting to note that NADH, right, is going to be the reduced form of NAD+. So NADH is basically just NAD plus electrons. You can think of it kind of conceptually like that. And there are H's that kind of just fit into this eventually, but we don't really care about the H's that much. We care about the fact that they're holding electrons. And FAD, the oxidized form of FADH2, is called FAD plus electrons, is going to give you FADH2. And these hydrogens are just kind of floating around. They usually come from the food, so like glucose, and the H's are just going to attach. They're floating everywhere. There's protons everywhere. And so, oh, by the way, H+, another word for H+, is proton, and I will make references to that later. So again, the important thing to note here is that NAD+ is the oxidized form of NADH, meaning that when NAD+ is reduced with an electron, right, it becomes NADH, and FAD is the oxidized form of FADH2, meaning that when we give an electron or give electrons to FAD, it will become FADH2. Okay? So again, just think of these as coin pouches that hold electrons, and these are empty coin pouches that do not have electrons. Cool? Okay, moving on. So, before we move on, I want to do a really quick check, even though I just said it, so hopefully, you should be able to answer this. Is NAD+ the oxidized or reduced version of NADH? So one more time, is NAD+ the reduced or oxidized version of NADH? The answer is NAD+ is the oxidized version of NADH because that means that it has a lack of electrons, or NADH is a reduced form of NAD+ because NADH is a form of NAD+ that has electrons. Wow, you can see how this could get really confusing. Alright, so that's important because, again, we want to be filling our money bags, our coin pouches with electrons, the entire process of cellular respiration. So this is going to be kind of the big picture of cellular respiration. There are 3 stages in total, and we'll be discussing where they happen, how they happen, and what's the point or why they're happening. So the 3 stages of cellular respiration, that giant equation of cellular respiration, which was C6H plus O6 plus O2 becoming CO2 plus H2O, remember that whole reverse photosynthesis equation? The 3 parts of that, kind of similar how photosynthesis had the light reactions in the Calvin cycle, the cellular respiration process has 3 processes within it, and that is glycolysis, the Krebs cycle, also known as, aka, the TCA cycle, also known as the citric acid cycle. So it has actually 3 different names. for historical purposes. And then finally, the last stage is called oxidative phosphorylation, which can actually be broken down into two additional substeps, and that is the electron transport chain and the process known as chemiosmosis, although a lot of people endearingly refer to it as simply ATP synthase, the main enzyme that's in charge of this process. And so really there's three stages of cellular respiration, and so you have glycolysis, you have Krebs cycle, and you have oxidative phosphorylation, and in oxidative phosphorylation you have these two processes also. One thing that I do want to mention is that you can get into extreme detail on all of these. In fact, if you are interested, I'd highly recommend you come into office hours, but we'll go over this in slight detail right now. One last thing I want to mention, actually, is that this process right here, oxidative phosphorylation, this process is where most of our ATP is made. This is where we're really going to cash in those coins, those electrons, those tokens, and produce loads of ATP. There is some ATP made in these two cycles, or these two processes, but really most of the ATP is going to be made in the third cycle. Steps 1 and 2 are mostly for filling our money bags. Glycolysis and Krebs cycle are both going to make more of our NADHs, and in fact Krebs cycle is going to give us NADH and FADH2. So we're really filling our token bags here in these two steps, and then in step 3 is our cash-in time, where we just like get a bunch of tickets, our ATP, so eventually we can use it to do work, buy prizes. You get the idea, hopefully. Okay, so before we go into the exact stages or the exact mechanisms of stages, it is important to note something called substrate versus oxidative phosphorylation. Now, substrate level phosphorylation, so this is substrate level phosphorylation versus oxidative phosphorylation is going to be kind of the bridge between making little ATP on the molecular side of things and making a lot of ATP on the kind of cellular respiration point of things. And what I mean by that is actually, if you see right here, substrate level phosphorylation is when you have one enzyme, like so, taking something that has a P or phosphate attached to it, and then this enzyme will actually take this ATP or take this P phosphate and put it onto this ADP to make ATP. This one ADP, right, taking one P and making ATP on this like one molecular level or one molecule level is going to be substrate level phosphorylation because it's taking the phosphate group from a substrate. And that substrate could be anything that has a phosphate group attached to it. Oxidative phosphorylation, on the other hand, is going to be when we're making loads of ATP all at once. And so really, the best analogy I can think of is when it makes it rain with tickets. It's like when you hit the jackpot on that one game where the light goes around in a circle and you have to make the light hit a certain spot and then tickets just fly out and you're like, woohoo! Yeah, oxidative phosphorylation. Going to be extremely important in that third stage, which is conveniently called oxidative phosphorylation, where we get most of our ATP in cellular respiration. Okay, so just think, substrate level phosphorylation is going to be making one ATP at a time, and then oxidative phosphorylation is making multiple ATP at a time, which is actually going to be around the order of 32 to 34-ish ATP per glucose molecule. Okay. Alright, so this is an overview, schematic overview of where things are happening and what's happening. And again, we'll talk about why. So glycolysis, that's spelled glycol. There should be a Y between the L and S right there. There should be a Y, glycolysis. That's interesting. So glycolysis is our first stage. In this stage, glucose is going to become something called pyruvate. In the process of doing this, we do get some ATP. We actually get 2 net ATP. So 2 ATP, is formed from this, and this is substrate level phosphorylation. It's small stuff, right? Little money. And so even though we do get some energy here, what's more important is we get NADH here. We get these coin pouches starting to be filled. Okay, and so we're starting to get our NADHs here, and then we move on to the citric acid cycle, which is stage 2. So this is stage 1, this is stage 2. Stage 2, the citric acid cycle also makes some ATP. We also only get 2 ATPs here. On the other hand, though, we do get actually 6 NADHs here, and we get 4 FADH2s here. So here, we've just filled up our money bags, right? We've got our tokens. And so our NADHs and FADH2s, we've got a bunch now. And so in glycolysis right here, we actually made 2 NADHs. In citric acid cycle, or Krebs cycle, it has a lot of names, remember, we're going to make more NADHs, more money pouches to fill with coins. And then we're also going to make these FADH2s. Voila. And so in total, just from these first 2 steps, we're actually going to make 8 NADH molecules, 4 FADH2 molecules, and then just counting the little money, 4 ATPs. Okay? So you'll notice in these first 2 stages, we've only actually created 4 ATPs, which is not a lot actually. We need actually millions and millions of ATP molecules. And so the real money is going to come again from these NADHs and FADH2s, okay, where they're going to be used in the process, the 3rd stage, oxidative phosphorylation, where we're going to take these NADHs, FADH2s, dump in the electrons, and eventually get out ATP. In fact, we'll get out about 32 to 34 ATPs. Okay, so in total, we'll end up getting around 36-ish ATPs from just one molecule of glucose. And in fact, this can be happening on like multiple parts of the mitochondria all at once. So one mitochondria will actually make a lot of ATP because cellular respiration would be happening on multiple locations on the mitochondria. Okay, so that's a kind of schematic overview. We're going to go into each step now. And this is where we're going to start getting a little bit detailed. Okay. So let's start with number 1, glycolysis. Okay, so we have glycolysis first. That is a C. Now, some of you may recognize the word lysis, right, and the word glyco from somewhere. So glyco actually has its, I'm trying to make that better, into a better C. So glyco actually has its roots in the term sugar, like glycogen is a complex sugar, okay? And lysis, right, remember in like hypotonic solutions, the cell would lyse, and in viruses, the lytic cycle lyse the cell. Lysis is splitting. Oh, and also photolysis and hydrolysis. A lot of lysis, right? Splitting, okay? That's supposed to be splitting. So, splitting. So, glycolysis actually quite literally means sugar splitting process. Splitting. Forgive my horrible handwriting. So glycolysis actually means sugar splitting. And glycolysis is really going to be taking this glucose molecule, this sugar, and catabolizing it, okay, or oxidizing it actually. We're taking electrons from it. So the process is actually a 10-step process that goes through multiple molecules, and evolutionarily, like those molecules, this reaction chain like set up over time because it was the most efficient manner. But basically, glucose, long story short, is going to end up becoming 2 pyruvate molecules. Okay, pyruvate. So pyruvate is going to be a 3-carbon compound that's going to be formed from glucose. Now, we don't really care about the pyruvate as much. Okay, and I'm actually going to rewrite pyruvate because that is awful. Oh man. Here's my pyruvate. So glucose is going to be broken down, this process known as glycolysis, and split into 2 pyruvate molecules. So if glucose is a 6-carbon compound, 4, 5, 6, and most of you know it as the ring structure, right? glucose is going to be broken down into 3 carbon compounds. So 2 3-carbon compounds. What's important about this, though, is not just that we're making pyruvate, but in this process of glycolysis, we're making 2 NADHs, right? And NADH, again, is this coenzyme that holds electrons, right? And we're also going to make 4 ATP. But actually, to go from glucose to pyruvate, we need to put into ATP. So we actually need to use up some energy to make this process go. And so our net product is going to be 2 ATP made in the end. Because even though we're making 4 ATP in the process, because we have to use up 2 ATP, in the end, going from glucose to pyruvate, the net ATP made is 2 ATP. So let's actually just kind of recap. In glycolysis again, we're going to be making 2 ATPs, 2 net ATPs, because we actually make 4, but we use up 2. And we're also going to be making 2 NADHs. Okay? Now, where is glycolysis happening? So to do this, we actually have to examine the anatomy of mitochondria and a little bit about the cell first. So we're going to draw some pictures. So this right here is a cell, right? It's got its phospholipid bilayer membrane. So this right here, if I zoomed up on this, you would see kind of this phospholipid arrangement that we talked about before, right? With its like protein channels and everything. So if I zoomed up on that, you would see the phospholipid bilayer, but I'm just going to represent it as one line here, because we just want to work with a microscopic. So this is a cell membrane. So this right here is my cell. Okay. Inside the cell, obviously, you have things like the nucleus, which holds the DNA and everything. What we're focusing on today is the mitochondria, and the mitochondria looks something like this. It's that poopy-looking thing we saw in the beginning of the lesson. If we look carefully, glycolysis is actually occurring in the cytoplasm of the cell. Oh my gosh, that's interesting. So glycolysis actually has almost nothing to do with the mitochondria, at least in terms of what it's doing or where it's doing it. It is going to be important for the mitochondria in terms of what it gives to the mitochondria, but in general, glycolysis happens... Ooh, I spelled glycolysis wrong. That is unfortunate. Glycolysis. Glycolysis happens in the cytoplasm. Another word for cytoplasm is actually cytosol. And this is actually more accurate because cytoplasm refers to the cytosol, which is just a jelly with all the organelles. And so cytosol is actually just the fluid, and then cytoplasm refers to the cytosol, the fluid, plus all the organelles like the lysosomes, the ER, and everything. So cytosol, cytoplasm, it's a technical point, but I will probably refer to the cytoplasm as cytosol now, just because that's cytosol. Sounds cool. Okay, so let's actually talk about the mitochondria now, because that's going to be extremely important for the next two processes. So this is my mitochondria. Okay. The mitochondria actually has 2 phospholipid bilayer membranes. So it has a double phospholipid bilayer membrane. I know, crazy. We think it's because it was just a bacteria that ate another bacteria one day. So mitochondria actually is going to have an outer membrane. So this right here is going to be my outer membrane. Okay? And then we're going to have a inner membrane. And these are both phospholipid bilayers. So this right here is going to be my inner membrane. Oh, look at that. Okay? And so this actually frees up this space right here. Oh, I swore I wouldn't use this marker again. This space frees up this space right here that I've just outlined, and I'm going to take away that because that was awful. And this space is going to be the intermembrane space now. and it's an actual physical space that things can hang out in. So between, that's a membrane, intermembrane space. Okay, so this space right here is actually going to be the intermembrane space. Okay, now this is a very simple drawing of the mitochondria. In reality, the mitochondria looks something more like this. So instead of drawing that simple kind of inner membrane like this, I've actually drawn what it more looks like, which is these squiggly lines. And so again, this is going to be the inner membrane, and this is going to be the outer membrane, and then this space right here is going to be the intermembrane space. So intermembrane space. Membrane space. Okay. So let's label a couple more things. Right here, this inside place is called the matrix. So this is called the mitochondrial matrix. It's the inner compartment. It's also known as the lumen. Okay. So this inside space right here, is going to be the mitochondrial matrix, or the lumen. Okay. And then again, this space right here is going to be the intermembrane space. So these are both places that molecules can hang out. And then finally we have the outer membrane and the inner membrane here. Okay? And the reason why this is important is because the Krebs cycle, or the citric acid cycle, is going to be happening in the matrix, and the electron transport chain is actually going to happen on the inner membranes. Okay? So we'll go into that now. Oops, one slide too much. So actually, let me just show you guys right here. Oops, too far. So if you see right here in the cytosol, ooh, I don't want that. In the cytosol, we have glycolysis happening. Inside the matrix, so this is a squiggly line, inside the matrix right here, we've got the citric acid cycle, or the Krebs cycle, happening, and then the oxidative phosphorylation part is actually going to happen along the inner membrane here. Okay, so hopefully that helps in kind of knowing where things are happening in the mitochondria. Now, let's talk about number 2, the Krebs cycle. So between steps 1 and 2, right, so in steps 1 we had glycolysis, in step 2 it's the Krebs cycle, and I'm going to refer to it as the Krebs cycle now, even though it has like 3 different names. What happened was, in glycolysis we were able to make pyruvate, along with our NADPH and our NADHs and our ATP. So pyruvate, before it even goes into the Krebs cycle, it actually has to become something called acetyl-CoA. And in the process of acetyl-CoA, it's actually going to create another NADH. And so this process has long been known as an intermediate step between glycolysis and the Krebs cycle. So the reason why that's important is step 2, that was an ugly 2. Step 2, otherwise known as the Krebs cycle, is basically the process of taking this acetyl-CoA and eventually getting 3 main products. Number 1 is going to be, let me try to find, okay. The number 1 is going to be NADH. Number 2 is going to be FADH2, and then number 3 is going to be, actually, surprisingly enough, ATP. So it does make some ATP. The acetyl-CoA does produce carbon dioxide in this process of the Krebs cycle. So the Krebs cycle does actually make some carbon dioxide too, but we don't really care about carbon dioxide that much. We're caring more about the NADH, FADH2, the money bags, and the actual energy we get. Okay, so this part right here, all I'm trying to kind of tell you is that pyruvate needs to become this intermediate acetyl-CoA in the mitochondrial matrix before it goes into the Krebs cycle. So if I go back, right here, this pyruvate right here is eventually going to become acetyl-CoA in order to enter the citric acid cycle, or the Krebs cycle. We're going to call this Krebs cycle. That's what most people know it as. Okay? So, let's keep going with the Krebs cycle. So this is a Krebs cycle. Okay. The Krebs cycle is actually a cycle. If we look at it particularly, let me actually find... So this is actually the Krebs cycle. And if you see right here, we have the acetyl-CoA, right, going into this very complicated step, even more complicated than the Calvin cycle. But what's important is that we're creating these byproducts, okay? So while we are creating carbon dioxide, again, we don't really care about carbon dioxide. What we are making is we are making NADHs, okay? We are making FADH2s, and we're also making ATPs, which is really, really good because, I mean, that's kind of what we want. And so in this entire process of making NADHs, FADH2s, and ATP, we are going to be eventually constructing our money bags for cellular respiration, or the last stage, oxidative phosphorylation. So again, this is kind of like a factory line where these molecules are just kind of processed, but what we really care about is what comes out of this process, which is, again, the NADHs and FADH2s. Okay? Okay. All right. So we're actually going to go back. Okay, so in the Krebs cycle, let's go over exactly what is being made as a byproduct. So, excuse me, gotta clear my throat. So in the Krebs cycle, the main products that are going to come out of the Krebs cycle are going to be from one... Oh, wait a second. That's odd. From one glucose, the Krebs cycle will actually give us 6 NADHs, 2 FADH2s, and in fact, I think earlier on this video I said 4 FADH2s in the overview. I'm sorry, that's actually 2 FADH2s. Yeah, okay, it's 2 FADH2s, 2 ATPs, and then actually it was supposed to be 4 carbon dioxide, but don't worry about the carbon dioxide actually. So this is actually what's going to be done in the Krebs cycle. In the Krebs cycle, in the mitochondrial matrix, inside the mitochondria, the Krebs cycle is going to eventually give us 6 NADHs, 2 FADH2s, and 2 ATPs. And the way it does that is, again, it's going to be taking... So we got our glucose becoming pyruvate, and then the pyruvate becomes acetyl-CoA, that intermediate step, and acetyl-CoA goes into here. In fact, because we get 2 pyruvates for every glucose, we have 2 acetyl-CoAs, and then each one gives us 3 NADHs, 1 FADH2s, and 1 ATPs, but if we double that, because we have 2 acetyl-CoAs, the important thing is, again, you just want to know that citric acid cycle gives you 6 NADHs and 2 FADH2s and 2 ATPs. Okay, so far we've gotten 2 ATP, 2 net ATP from glycolysis, we've gotten 2 ATP from citric acid cycle or the Krebs cycle, but here is where we really care. And remember, glycolysis also gave us 2 NADHs. And there was actually NADH also formed in this process here. But the key important fact is that now our NADH is able to go to the third and final stage, and we're going to finish with this one. Voila. And that is oxidative phosphorylation. So stay with me, we're almost done. Oxidative phosphorylation. Okay. So in oxidative phosphorylation, I should spell that better. So in oxidative phosphorylation, we actually have two steps. We have the electron transport chain, and we have chemiosmosis, or most people call it the ATP synthase process. And so there's a great video that's going to be posted at the end of this PowerPoint or this presentation that you can follow from NDSU Virtual Cell that really goes over this super well, but I'm going to try to do it justice with my chicken scratch writing. So with oxidative phosphorylation, the first stage, which is electron transport chain, so the electron transport chain is going to feature the inner membrane of the mitochondria. So this is the inner membrane. This is happening at the inner membrane of mitochondria. So on that mitochondria with its inner membrane like this electron transport chain is actually happening here. And actually, the second stage, ATP synthase, is also happening right here. Okay, so this is all happening along this membrane right now. So don't lose sight of that. Okay, it's all happening here on the mitochondria. So with that said, the inner membrane of the mitochondria is going to feature a series of proteins. So this is my inner membrane right now. It's going to feature a series of proteins, complexes, that are actually going to take in the electrons from the NADH and FADH2s. So just for perspective, this is the mitochondrial matrix, so this is the inner compartment where the Krebs cycle just happened, and this right here is the intermembrane space. Okay? And so right here, what's going to happen is we've just created all these NADHs and FADH2s. So NADH and FADH2 are actually going to give their electrons to this series of proteins. And these series of proteins is the electron transport chain. Okay? So NADH is going to give its electron here, and as it gives its electron, it's going to return to becoming NAD+, right? The oxidized form of NADH. And FADH2 does the same thing, except instead of giving it to the first protein, it gives it to the second protein. So it's going to give its electron here, and it's going to become FAD again. So full money bags giving the token, becoming empty money bag. Full money bag giving tokens, empty money bag. Okay? So right here in this electron transport chain, the first thing that needs to happen is NADH and FADH2 are going to give their electrons here. Now once the electrons are safely given, okay, what's going to happen is this very interesting phenomenon where, let me actually change colors, where hydrogen ions will be forced to pump across into the intermembrane space. And the reason why this happens is when the electrons bind to these proteins, they're actually going to want to jump to the next series. So these electrons, as soon as they reach their destination here, they jump to the next protein here, and they're going to jump to the next protein here, and they're going to jump to the next protein. And as each electron moves down, the way this entire mechanism works is these proteins are programmed, or they're meant to kind of pump hydrogen ions from the matrix area into the intermembrane space. And so, big picture idea is that as electrons move down the chain, what's going to be happening is that action is coupled with pumping hydrogen ions into the intermembrane space. So over time, as NADH and FADH2 keeps bringing electrons here and keeps making these electrons jump down, these positively charged H pluses are going to be pumped across. Now a great question to ask is, why does H plus get pumped in the first place? That has a little bit to do with the organic chemistry molecular structures of the protein channels, or the proteins that are on here. But again, watch the NDSU video, it's very good for this. So, big picture, okay, in the electron transport chain, in the ETS, or ETC, electron transport chain, many hydrogen ions, okay, H pluses, are pumped into the intermembrane space. And this was only possible because of the electrons that were kind of dumped down the chain. Okay? And so, once we have this electron transport chain making a bunch of H pluses, we move on to our second step. Okay? And our second step is going to be the ATP synthase. So, let's talk about what's happening in ATP synthase now. Or chemiosmosis, that's also what it's called. So right now, along the inner membrane of the mitochondria, so this is my mitochondria, I know it's not the prettiest mitochondria. So right now, on the mitochondria, due to the electron transport chain pumping hydrogens into this space right here, so now we have a lot of hydrogens, we're actually going to build up a huge hydrogen concentration here. Okay? And so we have all this hydrogen ion, these hydrogen ions, and another word for, again, hydrogen ion is proton. So we have all these protons here, hydrogen ion, proton, same thing. So hydrogen ions and protons are all kind of building up here because of these electron transport chains that are pumping hydrogens into the intermembrane space. So now that we have a lot of hydrogen ions here, due to the laws of diffusion, right, these hydrogen ions that are in high concentration in the intermembrane space are going to want to come back into this matrix, the mitochondrial matrix. This is just the same diffusion we saw with osmosis, where things are going from high to low concentration. So these hydrogen ions are going into the matrix, and as they go into the matrix, they're going to pass through this rotor-like thing. And this rotor is possibly one of the coolest things ever. These rotors are possibly one of the coolest things ever, so if you see right here, this is actually the intermembrane space. I know this video is going really long. It'll be done soon. The intermembrane space, and this is the matrix. What's going to happen is these hydrogen ions are going to actually physically flow through this molecule right here. This will spin. It's awesome. It's a rotor. It's a rotor in your organelle. Like, before motors were made, our cells had motors. Like, what the heck? Okay, so anyways. So this rotor is actually going to rotate, physically rotate when these hydrogen ions go in. That's going to activate this knob right here that's in the matrix. And this matrix's main job, so this knob, this catalytic knob's main job, is actually going to be to take ADP and phosphate and catalyze the reaction of making ATP. And that's how we make a bunch of ATP. So this is actually where we make most of our ATP. So one more time, the hydrogen ions are in the intermembrane space, are going to flow through this rotor and come back into the matrix, but in the process they're going to activate this beautiful molecule known as ATP synthase, and as this thing is activated, it's going to take ADP and PI, this phosphate, and make our ATP. And in fact, it is this process right here that's going to give us about 32 to 34 ATP. Okay? So, just to recap, final summary, glycolysis, which is happening in the cytosol, or cytoplasm of the cell, is going to give us NADH, okay, 2 NADH, along with 2 ATP. This right here, in the process of going from pyruvate to acetyl-CoA, that also gives us NADH. This acetyl-CoA will go into the Krebs cycle and give us 2 ATP, but also it'll give us 6 NADH and FADH2. So all of these are being made from citric acid cycle. Finally, all the NADHs that were made from before are going to go into this final stage, oxidative phosphorylation, where first off, these drop off electrons that will pump hydrogens into this space right here, these protons into this space, and these protons will go down through the ATP synthase molecule, that cool rotor molecule here. All right, these hydrogen ions will go down and then give us our ATP by activating that enzyme. And so this will finally give us about 2 plus 2 plus 32 or 34 ATP, or in total about 36 or 38 ATP per glucose molecule. And one glucose molecule is not that much, because remember, one starch molecule, right, when we're eating like flour and bread, has millions and millions of glucose molecules, like in a chain. I don't know if millions is the right number, but you know, a lot of glucose molecules. So, in total, that's how we get most of our energy from glucose, and that's also why glucose is a very quickly metabolized sugar. But just for reference, it'd be great if you could go on NDSU Virtual Cell, look up cellular respiration photosynthesis. Actually, you could look at photosynthesis too, but Go look at electron transport chain. I will post it on below this video. And Crash Course Biology with Hank Green has an excellent overview of the entire process too, and hopefully that will help you. Okay. With that said, take the Emoto quiz, write your comment below. Again, I'm so sorry this video ran long. Thankfully you have no school on Monday. Woohoo. And I will see you guys on Tuesday and Wednesday, and we'll have actually a cellular respiration obstacle course. It'll be pretty intense, cognitively, not as physically, and we'll be like running through the matrix and stuff. But we'll do that when we come back to class this week, next week. This week, Saturday part of this. Okay, we're done.